Mark, and we will have time for discussion in not too long from now. Next up is Aris Baras from Regeneron Pharmaceuticals, where he's the vice president and also the co-head of the Regeneron Genetics Center. Thanks, Aris.
Thank you all, to Jeff, Russ, Nadim, everyone's very grateful for the opportunity for Regeneron to participate, and I'll dive right into it. Absolutely. So rehashing a little bit what Nadim already threw out, which is, drug development is hard. It takes a long time. It's risky and costly. And over the last several decades, the industry has had a lot of innovations from small molecules to biologics, and we've addressed bottleneck after bottleneck to try to make this process more efficient and have a higher likelihood of success. I think one of the points to appreciate is that one of the remaining bottlenecks, one of the big ones, is drug targets, well-validated drug targets. So I pulled down some facts from some pretty good studies that had a lot of good methods and data they were pulling from, as well as some of our internal databases. And collectively, in our industry, we're all pursuing roughly the same 500 to 1,000 genes or so. And this is coming from products that are approved and things that are in development. Earlier than that, it's hard to tell what people are pursuing and what the drug targets may be. But as you all appreciate, this is less than 5% of the genes in the genome. So we've got a lot of work to do to understand what these genes are doing, what their function is, and a whole host of other genes that we haven't yet thoroughly explored. I think human genetics has proven itself to be a validated approach to optimizing and improving our drug development processes and getting at these well-validated targets. And I can point to some of the early pioneers and successes, many sitting in this room, from NIH-sponsored efforts with the sequencing centers and the NCI, Kari and his work with Decode Genetics, Mark and the work they've been doing at the Broad, and many, many others across this community, many times coming from a single investigator or a small group studying a rare disease or unlocking some major insights. And I think this all has us convinced, obviously, that human genetics supporting these drug targets or various links we make to potential indications we pursue is absolutely an approach to take. And we believe, and I think hopefully many here, that you can't do modern day drug discovery if you're not incorporating human genetics into your approaches. So this is just selected examples of some drugs, many of them coming from human genetics insights, then being translated into actual successful therapeutics development. And some of them are after the fact, taking a look and noticing, oh yeah, there was very predictable genetics there that would have led us to predict the pharmacologic effects. And importantly, I'm not looking at the GWAS and other types of studies, which also provided many insights, but these are loss of function mutations or Mendelian presentations. And the point is, there's maybe a couple more tables like this, but that's it. And so, as Nadim mentioned, we have the resources and tools now to hopefully do this beyond onesies and twosies and over decades, but now to do this rapidly and repeatedly over and over again and really transform the way we do drug development and bringing new medicines forward to patients. So this is our simple view of how we want to apply human genetics to our approaches. At the top there is target discovery. At a company like Regeneron, we're pursuing 200 different targets, and few of them have human genetics support behind them. So they come from mouse genetics, molecular biology, biochemistry, even looking at some other clinical pharmacology that's gone on before us. And so human genetics can really provide these human experiments that can teach us a lot about validating a target. We also, on the flip side of that coin, is looking at indication discovery. So the existing targets we have and programs we have, we want to use human genetics to help guide us to the indications where there's a clear link, or expand our indications. And lastly, looking at biomarkers and using pharmacogenetics to identify patients who may be more responsive or to avoid exposures when someone may have an adverse effect that we can predict. Okay, so behind those goals, how do we go about building a program that would support all of this? So many of the things that you heard from Joe and Mark were key tenets of how we wanted to build our program. And so one of the most important ones was that we believe in an integrated approach across genetic architectures and going through general population strategies, and our foundational cornerstone effort there is with the Geisinger Health System that I'll tell you a lot about. But we also very much believe in Mendelian genetics, really traditional approaches to genetics. And we have thousands of families that we've sequenced as part of that effort with leading collaborators like Wendy Chung at Columbia University, Jim Lepsky at Baylor and other places we've been working on Mendelian genetics. And then, of course, you'll hear from, really the leader in this field of founder populations, Kari Stephenson. We've been working with groups in the Amish and Mennonite, where my colleague, Alan Schuldiner, has done phenomenal work there, as well as we're working in Founder, Quebec. And lastly, to Mark's point, we also appreciate that approach like Geisinger or a general population approach, you get into lower prevalence diseases, less than 1% population prevalence or so, and there are many, many diseases like that. We're just not going to be powered, whether it's Geisinger or the PMI. And so we're having a lot of luck, and I'll give you some examples looking at cardiometabolic diseases, asthma, COPD, some allergic conditions. There is a large list of things, obviously, that are common and prevalent and we can attack with the tools that we have. But we also supplement our approach with a lot of these disease-specific cohorts where there are huge numbers of individuals who have already been ascertained, and also they can be more deeply phenotyped with measures that are, and related traits that are important for that disease and understanding the mechanisms. So jumping into Geisinger, I think Joe gave a nice introduction of what a community type of regional health system, some of the characteristics. And for Geisinger, it's quite similar, if I could say, and it's in central Pennsylvania area. There's a couple million individuals who are part of that catchment area. And being someone from New York, it's a perfect example to give where we have 5 different providers for all of our healthcare. And it's the exact opposite down in Geisinger, where you have essentially almost 98% of all the individuals that I just cited in their catchment area, all their encounters are captured in that health system. Furthermore, they've had electronic health records since the early 90s and have been amongst the leaders in terms of analytics using that data. So I will show you some data here about just how much the high density and longitudinal aspect of this data and how important it is for genetic discovery. So I've cited here just a few of some of the data elements we pull out in terms of the number of encounters per year, our sequenced cohort of 60,000 individuals, exome sequenced and whole genome array genotype at Geisinger have on average about 15 years of longitudinal data, and that's about 10 when you go out to the broader health system. And there are just hundreds of thousands of individuals with each one of these labs and various diagnoses listed on the bottom there. And just like genomic data that requires a lot of analysis, filtering, and curation, it's really important that you have those methods in place for phenotype data as well. And I think this will be a key challenge for the PMI. And we've spent a lot of time, and Geisinger's efforts certainly predate us working with them in curating this data and really cleaning it and prepping it and having well-defined algorithms for various phenotypes and having longitudinal measures for a lot of these quantitative traits that we use. So switching now to back to the genetics, we at Regeneron focus on rare, large effect coding variation. For us, drug company, most of what we go after is gene products, so that makes sense to go after, to look at those types of mutations. And those are the things also that are highly interpretable to us. And one class, as an example, is looking at predicted loss of function variation. And whereas each individual may have approximately 20 loss of function variants using some pretty stringent filters, when you roll that up across the population, you actually see here quite good coverage of almost, more than 90% of genes have at least one individual with an LOF. And it's obviously much less when you get to homozygous instances of that. But as we've seen, and Mark and others showed you, you really need hundreds of these individuals. And so we need to take that bottom curve and push it way up to really be powered to understand the consequences of these mutations across many diseases. And so I think that's where it's going to require us going yet another magnitude of order and scale and going to places like founder populations where you may have by drift some of these instances enriched and you can really study them. So I'm going to highlight here a few examples of our discovery work using these types of resources. So to give you some background, and we do this essentially on every target and every clinical program we have, and I mentioned roughly 200 of those we're working on. We have a late-stage antibody that's targeting the angiopoietin-like 3 gene product. And it's in late-stage development for dyslipidemias, recently got orphan designation status for familial hypercholesterolemia. And we've also been studying a related family member, ANGPTL4. Both of these are natural or endogenous inhibitors of the lipoprotein lipase molecule, which mediates triglyceride metabolism and triglyceride-rich lipoproteins. Some of the background here, work from the Dallas Heart Study, as well as from Say-Catharacein and family-based studies, really implicated these genes some years ago in terms of being very important in modulating lipid levels. But what hadn't been established yet is the association with clinical endpoints. And in the case of ANGPTL3, you do have an LDL effect, but in the case of ANGPTL4, you don't, or at least we don't see one, and others haven't seen one. So there are other lipid fractions involved here, and the question was, would there be an effect in terms of loss of function mutations and effect on things like coronary artery disease? So on the left there, I show a few tables that we had from a recent publication that our team put out. There was also a back-to-back work that was done by Nate Steitziel and a large group of collaborators, basically identical findings, really. And this was looking at the ANGPTL4 gene. Two sets of variants were looked at. One was a low-frequency variant, coding variant, E40K, which was well studied and had been molecularly characterized to have some inactivating properties, I won't say complete null. And this was shown, as you see in the bottom there, to have roughly a 15% reduction in the odds of coronary artery disease. And this allele is present in about 2% of the population of Geisinger and other similar populations. What both our groups found were that loss of function mutations, so now we're quite sure that we've got a null. And our most common null, we did a lot of experiments to show that, in fact, it's not secreted, it is a true null. We found more pronounced effects, roughly halving of odds for coronary artery disease. And this is work, as I mentioned, that was published just about a month ago. We also found, which is very interesting, and this is now being written up and hope to be published soon, an effect on type 2 diabetes as well. And this was similar magnitude of effect in terms of protecting from type 2 diabetes. And we're very interested in that mechanism, how that's working. And coming back now to ANGPTL3, the molecule that's in late stage development, we're doing some early work on that, and some of the early findings are suggestive. We need to replicate and confirm these things, that there might be a protective effect there as well. So now switching gears and showing you one of the other approaches, which is the classical, more Mendelian approach to things. We've been working with Wendy Chung at Columbia University, and she and Rudy Leibel over the last decade or two have done a phenomenal job collecting these large pedigrees in half a dozen different cardiometabolic disorders with real kind of evidence of Mendelian inheritance from all over the world being seen in New York City. And I cite here an example of pulmonary arterial hypertension. One, it's extremely interesting to us. I think there's been a lot of progress there in the last decade in terms of therapeutic development, but still there is a huge amount of morbidity and mortality associated with that. And importantly, only about 5 or 6 genes have ever been discovered and associated with that. So you see the details there in terms of the number of families and then additional singletons that were available for confirmation. And what was unique was this was enriched for pediatric onset, where you usually have onset in your 30s or 40s for classical PAH. And we identified a new gene there quite convincingly, 16 different cases from families and single probands where we found multiple hits in that gene. So the last story I'll tell is, and we are huge believers in this at Regeneron, is that human genetics is unbelievably important, and it has to be considered as a piece of the puzzle. And so I think one of the things we'd love to impress upon everyone from our perspective is that marrying these with functional biology and with clinical pharmacology is a necessary equation in terms of how do we take the genetic insights and translate those or convert them, as Mark had said, to real drug discovery opportunities and therapeutics that can get to patients. And so the genetic discoveries behind FOP, a very rare disease that is characterized by heterotopic ossification or bone formation of soft tissues like skeletal muscle, tendons, that leads to basically severe disability and death, oftentimes due to asphyxiation of thoracic cavity and a fixed rib cage. The genetics behind ACVR1 are well established, and that was not done by us. But what's the mechanism behind all of this? To my colleague, Aris Baras, no surprise, another Greek at Regeneron, was looking at all of the known ligands for this BMP receptor and basically just looking at the difference between the mutant receptor and the wild-type receptor and signaling. And what he, in a nutshell, what he found was that activin A, one of those ligands, only, and that's one of the mutations found in patients, put into a genetic model, we knocked in that variant, only activin A induced heterotopic ossification in the mutant receptor and not the wild type. And further, when you use an activin A neutralizing antibody, you basically abrogate the heterotopic ossification. This is something that we're working now to develop in clinical development. So I summarized here the guiding principles, and then some of the challenges are really the same that others have already mentioned in terms of we really need large study sizes, we need collaboration, we need to do some more work in terms of understanding rare missense variation and perhaps even non-coding variation as well. And we have to have these opportunities to pursue the biology and the clinical pharmacology downstream of these human genetics discoveries. Thank you all.
That was terrific, Aris. Thank you. So we have a short break now. We're going to come back here at 9:45 when we'll have our last two speakers and a panel discussion to follow. Please be back at 9:45.